Let's talk about the estimation of protein in foods. While this analysis is done actually for multiple purposes and different reasons, I think one of the most important thing to consider for determination of protein is actually to look at the biological activity of proteins. As you know that most of the proteins are enzymes in nature, so we need to know their activity. Like they have got many potential application in food. For example, we can talk about certain enzymes such as meat tenderization enzymes. So we need to know the activity of enzymes in our final products. Similarly, we can also know the ripening enzyme present within fruits and their activity, and same can be applied to trypsin inhibitors in different vegetables or pulses. So in order to know the activity of enzymes, because enzymes are basically protein, we do this analysis of protein analysis. Then we also need to know the functional properties of food. Like most of the proteins, they do impart functional properties or functional characteristics to certain foods. For example, if you talk about gluten contents in wheat, while gluten content has gliadin and glutenin content, it has got very important implication in the formation of bread or all the bakery products. So in order to know that functional property of bread, we need to know how much gluten content is there, and within gluten how much glutenin and gliadin are there. Similarly, in case of milk as well, for example, from milk we made certain products like cheese or yogurt. So we need to understand the behavior of casein contents within the milk so that it can be converted into cheese. So casein is basically a protein, and that functional property of protein is determined by various kind of protein analysis. One another most important reason for why this is routinely done is nutritional labeling. As you see that most of the food products would have nutritional labeling, and they would mention the amount of protein present in a food. So in order to mention that accurately, we need to do the analysis first, and then that can be labeled on the product. Why we do this food protein analysis is there could be different scenarios. Like in certain cases you want to know the total protein contents, like how much protein is present in a specific food, or the amino acid present, or there's the sequence of amino acid, how many amino acids are there, what is the relative proportion of that. In certain cases in a mixture, if you want to know how much protein is there within that mixture. And we also want to know that during different extractions or purification procedures, like how much protein actually are we extracting from the substrate. So there could be all different reasons why we do this food analysis. Sometimes you also want to know that what are the non-protein contents within a food. So that also can be indirectly derived from the food protein analysis. Similarly, nutritional value, digestibility of proteins, their absorption, their bioavailability, or protein efficiency ratio. These are all the concepts that we earlier discussed as well. These are all the reasons why protein analysis can be done in food. And I think one of the most important and widely used method is Geldol method to determine total protein content in food. This is very old method developed by Geldol in 1883, and this has remained the very powerful tool to analyze total protein content since then. Although there have been changes in different, the method has remained same, but the changes were only in the equipments. But the basic principles always remained same since 1883. The protein contents actually have got, this protein determination have got 3 steps. There are these 3 steps involved, and these 3 steps are digestion. In this step we digest the food so that the other compounding factor, other confounding factor which are there in the food, they are actually get rid of. Then in the second process we do distillation. In the digestion process we release ammonia. The protein contents are determined by nitrogen, and nitrogen is converted during the digestion process into ammonia. So this ammonia is then distilled into another solvent, and that total nitrogen actually is released from the food in the form of ammonia. This is distilled. Once all nitrogen is distilled, this is then titrated against an acid. Yes, if you look at this Geldol apparatus, it consists of these three units as we discussed. One is the flask in which we add food sample, and then there is a heating element. So this is heated. The food is heated in the digestion flask, where we do add certain other reagents as well. So once it is heated, the ammonia compound goes out of the flask into this condenser. So this condenser have got a water inlet and water outlet that actually reduces the temperature of this condenser unit. So nitrogen content, where this comes in the form of a vapor, it becomes liquid here, and then from there it reaches to this flask that would trap all the ammonia. So you can see these little dots as ammonia gas bubbles. So this would be dissolved in this flask. So this is overall the principle of Geldol apparatus. So it has got three units: digestion, distillation, and the collection of the ammonia gas that eventually is titrated in the third step. So as we discussed, that protein is digested with sulfuric acid. That's a strong acid, so that would digest all the food into in the form of ammonia. And this the other compounds which are produced are carbon dioxide and water that do remain within the flask. As we discussed, this digestion flask is distilled into condenser, and this ammonia is trapped into the final boric acid solution that would capture. Boric acid is basically a weak acid that would be reacting with ammonia, and it would bind or absorb all the ammonia within the boric acid solution. And this boric acid solution then is again titrated against another acid just to know that how much ammonia was there. So basically all the ammonia, when it goes into the flask, this ammonia makes it basic in nature. We titrate against an acid. That acid would neutralize all the ammonia, and once the ammonia is neutralized, the color of the solution would change due to the indicator, and that would indicate that the reaction has been completed. So from that onward, we do calculate how much nitrogen was released from the food, and that nitrogen is multiplied with a multiplication factor to know how much protein was there in the food. So this was the overall process for the protein determination in foods.